Hey, here we are. So this is a video about the structures on the cell surface. So there are a lot of structures in bacterial cells from lecture 2, and you should know them, but the ones that are most important are the ones on the surface, the ones that make up the outer parts of the cell because those add the structure to the cell, and a lot of times those are the ones that we attack when we're trying to kill microorganisms. So antibiotics attack the outer parts of the cell. Disinfectants attack the outer parts of the cell. Hand sanitizer attacks the outer parts of the cell. So we really need to know about those. So let's review what a cell membrane looks like. So I don't know if you've ever seen this diagram before, but you should recognize what this is trying to show you. This is a cutaway view of a generalized cell membrane. So what would these little spherical things be? And what would these little tail things be? Why don't you pause the video and tell me? So those are phospholipids. These are the heads that are hydrophilic, so they interact well with water outside the cell or inside the cell. And these are the tails that are hydrophobic. They repel water and they interact with each other instead, and they make the inside part of this membrane more or less water resistant, we could say, almost waterproof. So this is a really good barrier. It's going to prevent any charged molecules are not going to be able to cross this. Any large molecules like proteins, which are also charged, they could never cross this membrane unless there's some kind of transport protein that lets them through, or a channel. Also, phospholipid membranes are flexible and floppy. They are very similar to soap bubbles. They're kind of put together exactly the same way, and they're about as strong. They're not very strong, but they're meant to be flexible. Bacteria also have cell walls made of peptidoglycan, which is a strong polymer. And if the cell wall breaks, the cell dies because the cell membrane isn't strong enough to hold it together. So let's draw a typical bacterial cell. I think this will help. So I don't know. This software has been updated since I last used it. Ooh, that's cool. Let's try Galaxy. So we're going to draw a typical cell. So on the inside, it's going to have DNA, proteins, water, other stuff like that. And then surrounding that, generally speaking, is going to be the membrane. So that's going to be a phospholipid bilayer. And all cells have this, whether it's an animal cell, a bacterial cell, a fungal cell, a protist cell. Cells have a cell membrane made of phospholipids. It's a really, really good system for keeping the inner parts in and the outer parts out. You may remember a term, selective permeability, that describes one of the things phospholipid bilayers give us. So what bacteria also have is some kind of a cell wall made of peptidoglycan. And ooh, let's try rainbow. That's cool. So the cell wall is on the outside of the cell membrane. And there are different types of cell walls. So I'm going to draw the different types kind of one at a time here. But just generally speaking, this is going to be the cell wall. You know what I've noticed? I'll tell you, Galaxy and Rainbow look pretty similar. So, whatever. So let's look at some specific things. So the cell wall is made of, generally, peptidoglycan. So that is a polymer. And the cell membrane is made of phospholipids. Now they have different jobs. The cell wall and the cell membrane have different jobs. The cell membrane keeps the cell waterproof. The cell wall makes the cell strong. I'm going to post a video that I made in the past where I show sort of how this is supposed to work using a balloon and a sock. But for now, what you could understand is that if you have water out here water is going to tend to rush into the cell because there's all this stuff dissolved in the water in the cell, and just osmosis is going to pull water in towards where there's a bunch of stuff dissolved. So that means there's pressure always pushing out. Oops, yikes. Always pushing out. Pressure pushes out. And a cell membrane is weak, and it would keep expanding till it popped. But a cell wall is not. It can't expand. It's like a net. It's like a giant spherical molecule. It can't get any bigger, and so it limits the size of the cell and allows the cell to have this high pressure without popping. Okay, so let's look at the different components. So the cell wall, well, no, let's go back. Let's draw this out. Okay, so that's a cell. Okay, there are two different types of cells we want to think about here. The first one we're going to call gram-positive. Now these cells are very common. These are a type of bacteria, and they're very common. And how they're designed is they have a membrane with DNA and stuff inside it. And then they have a very thick layer of peptidoglycan around them. Very thick layer, very thick cell wall. Like it might be, like, relatively speaking, this thick. Very strong, very thick peptidoglycan layer. Now it's also going to have some structures in it you should know. I'm going to draw as little red lines. These are... Okay, so you use a new drawing program and it turns out it just draws arrows automatically. That's cool, but that's not what we wanted. I want to draw what's called teichoic acids. Now these physically connect the gram-positive membrane to the gram-positive cell wall. So these are teichoic acids. A gram-positive cell is also going to need to have a lot of transport proteins. What? No, yellow. Transport proteins. There's going to be a lot of proteins embedded in the cell wall. Now, this cell wall, if we go back to the cell wall, something I should have mentioned before, we go back up here to the cell wall. The cell wall is permeable. It's not supposed to block anything from coming in and out. It is supposed to let proteins in and out. It lets ions in and out. The cell wall is like a net. It's a mesh. It's porous. The cell membrane is not. The cell membrane is supposed to be waterproof, more or less. So what you have is this waterproof layer, keeping the inside of the cell in, and then a thicker kind of mesh net layer on the outside. So that's gram-positive. Now let's look at gram-negative, and these are a little bit more complicated. So gram-negative. And the way these look is they have a cell with DNA and stuff. DNA and stuff inside. And then they have, outside, they have a thin layer of peptidoglycan. So, this is membrane, peptidoglycan, and then outside of that, they have another membrane. So they actually have two membranes, but the membranes are different. This is the outer membrane. So the inner membrane is the real membrane for the cell. This is the cell kind of ends at the membrane. Then the peptidoglycan is that structure that gives it some strength, and then the outer membrane has other features to it. So the outer membrane kind of camouflages the cell, but it also defines Yeah, that should be good. It also defines this whole region between the two membranes. This whole area between the two membranes is the periplasmic space, or the periplasm. Periplasmic space, or the periplasm, is this blue area between the cell membrane and the outer membrane. And that periplasmic space is a safe place for the cell to keep stuff outside. So imagine a porch, or a screened-in porch, where you can keep some of your furniture and some other stuff, and it's not going to get rained on, and that stuff won't float away. The cell can keep proteins that need to be outside of the cell in the periplasm, and they are basically in the outer environment. Because the outer membrane is porous. Because it has big proteins called porins that let stuff through. Porins. Those are proteins that let molecules cross the outer membrane. It's not meant to be waterproof, but it's meant to be, it's meant to slow down anything that might leave or come in. And so the periplasmic space ends up being a great place for gram-negative cells to perform chemical reactions that might be dangerous to do inside the cell, without losing the proteins that they put out there for that purpose. And the porins in the outer membrane let molecules come in and out. And then the cell membrane itself, which separates the inside of the cell from the outside, is going to have all of the transport proteins, all the self-surface receptors that any other cell would have. But it's inside this protection. It's inside the periplasmic space. It's inside the cell wall. It's safe in there. So now that I've drawn this out, I want to look at the diagrams in the slides because they'll make a little bit more sense once you know what's coming. So just to review, a gram-positive cell is going to have a cell membrane and a thick peptidoglycan layer, and then teichoic acids holding it together. Gram-negative cell is going to have a cell membrane, a very thin gram-positive layer, another membrane, which is porous because it has these porin these porin proteins, and then the space in between the two membranes is called the periplasm, or the periplasmic space. There's one other thing about gram-negative cells that I probably should draw in here, and I shouldn't have skipped. So let's find a new color, a new really exciting color, and make it like a 4. Some of the outer part of the outer membrane is made of a specific lipid attached to a carbohydrate, attached to a protein. In some cases, it's called lipopolysaccharide. So most often it's a lipid attached to a carbohydrate or sugar. So let's go back to the slides and see what all of this is. So this is a cutaway view of a gram-positive cell membrane and wall and a gram-negative cell membrane and wall. This is showing a lipid bilayer. So these spherical things are meant to be the phosphates and then the little tiny tails in between. And both cells have pretty much the same kind of cell membrane. And then a gram-positive has a thick peptidoglycan mesh. Gram-negative cell has a thin peptidoglycan mesh. The gram-positive cell has these teichoic acids sticking out. The gram-negative has different proteins that we haven't named. And then the outer layer of the outer membrane has special lipids called lipopolysaccharide. Here is a slide that distinguishes peptidoglycan from the cell membrane, things I've just told you. This just describes gram-positive versus gram-negative. And then here's another diagram of a gram-positive cell. What I just mentioned. And here's a gram-negative cell. when you see different diagrams of the same thing drawn in very different ways, that is a really good learning opportunity for you, because you can look for what is common between those drawings and what is different between them. And that's a very good exercise for your brain to help you with understanding. So then there are some slides that just walk you through what the different structures are. And one very important one is LPS, or lipopolysaccharide. Lipopolysaccharide is also known as endotoxin, and that's because it is detected by our immune systems, and the immune system responds quickly by triggering fever and inflammation. So that means that if this cell gets into your blood, and your immune system detects the lipids on the outside of a gram-negative cell, the immune system will respond very quickly with fever and inflammation. That's going to happen even if this cell is dead. So this is one reason why it's dangerous to inject things into humans, and why if you're going to inject something into a human, you have to get it from a pharmaceutical company that could sterilize the water and test it to make sure there are no dead gram-negative bacteria in it. Any water you get that will be injected into a human has to be tested for the presence of endotoxin. If it has any endotoxin, it can never be injected into a human, regardless of how pure it is in other ways. So that's the cell wall, cell membrane. These are the structures of the cells. If I ask you to draw a diagram of a cell and it's gram-negative or gram-positive, that's the level of detail you need. And I like this kind of drawing because it's very conceptual, and it helps us think about the functions of these things instead of really their detailed structure. I do recommend you watch the video with the sock and the balloon. That video also has other topics in it. I made it, 2 years ago, but it's still worth looking at. Okay, well, thanks for your attention to this. Now you know the central essential parts of cell structure. You're going to have to look through the slides and look at the other structures that appear in cells, but these are the most important. These are what really defines gram-negative and gram-positive cells, and the vast majority of bacterial cells you're going to think about are either gram-positive or gram-negative. Cool. All right, I will see you soon.